Okay, shall we start? Good afternoon, ladies and gentlemen. Welcome to the ANZ SNM Radio Pharmaceutical Science and Combined SRS session number 2. Let me introduce you to our first speaker this afternoon, Dr. Weibo Kai. He needs no introduction. He's a pioneer in his field and Associate Professor of Radiology and Medical Physics, Bioengineering and Material Science and Pharmaceutical Sciences at Wisconsin, USA. Amongst other accolades, Dr. Kai has authored over 240 peer-reviewed articles, more than 20 books and chapters, and he's also served on the editorial board for scientific journals, Theranostic Journal and Nuclear Medicine. Without further ado, let's welcome Dr. Weibo.
Thank you. Is this on? Is this one on? Okay. Well, thank you very much for the kind introduction, Device, and also to Vijay for the invitation. And today I'm going to show you a little bit of what we did over the last 10 years or so on imaging and imaging as a tool for better for better therapy, kind of the precision medicine related work. Okay, so our group is mostly focused on 3 different areas: the development of new imaging agents, mostly based on PET, position emission tomography. We also test a variety of different nanomaterials for different kind of applications: imaging, image-guided drug delivery, and therapy. The goal of imaging and nanotechnology is mostly for targeted therapy for precision medicine. Our main focus is on cancer, but we are working on molecular imaging. It's focusing on certain molecules and say certain overexpressed receptors. So the same agent could be used in other diseases as well if you find the right target. So today's talk will be divided into three parts. I'll give you a little bit of introduction, maybe 5 minutes, and then I'll show you a few examples of the imaging agents that we have developed, mostly focusing on PET, but also on multimodal imaging in cancer and in a few other diseases. Then I'm going to spend maybe 10 minutes on the nanoparticle-related work, using imaging as a tool to better treat cancer and as well as a few other diseases. So this is the hallmarks of cancer. I think you're all very familiar with this. One of the hallmarks is angiogenesis, which is the formation of new blood vessels from the already existing blood vessels. During this process, there are a lot of different proteins or other cell surface receptors that could play a role. This a reviewer wrote a few years ago. On the outside are some of the targets that are being imaged with different kind of targeting ligands: peptides, proteins, antibodies, antibody fragments. In that review, we cover 3 different diseases: cancer, cardiovascular disease, and chronic inflammation. And in our group, we are mostly focused on 3 of those targets: integrin alpha beta 3, which we use RGD peptide as a targeting ligand. We can also use VEGF to target the VEGF receptors, or we can use antibodies to target either of the VEGF or VEGFR. We have also spent quite some effort on targeting CD105, which is another target that has been rarely studied by other groups. And CD105 is also called endoglin. It's a cell surface protein. It's a co-receptor of the TGF-beta receptor. What's really important is that the expression of this protein is highly correlated with kind of the aggressiveness of different solid tumor types. So in many different solid tumor types, the expression of CD105 is correlated with poor survival. So it's a very broad spectrum target, and we can use the same agent for targeting many different solid tumor types. The expression is also very specific. It's only on the proliferating endothelial cells, but not on the mature vessels. The expression level is also very high. It could be a few million copies per cell. So it's a very attractive target for targeting and for imaging as well as for therapeutic applications. So this is the data from a review article. If you stain for CD31 on the left are the normal tissue, on the right are the tumor tissue. If you stain for CD31, you see staining in both tissues, indicating that CD31 is expressed on the vasculature of both normal vasculature, mature vasculature, and the tumor neovasculature. While CD105 don't have any staining in the normal tissue, it's only in the tumor tissue. So it's a much more specific target for tumor active angiogenesis. So that's a brief introduction, and for the imaging agents that we have developed, I'll show you a few examples of CD105-based work. For this, we use TRC105, which is an antibody that binds to both human and murine CD105. That's really important because when we inject tumor cells into mouse models, the vasculature is of the murine origin, so the antibody has to bind the murine origin if you're imaging the vasculature. The antibody itself is already in phase 3 trial now in the United States, so hopefully in a few years it could be approved by FDA. as a new anti-cancer drug in combination with some of the other therapeutic agents. Cetuximab is a perfect control because it's the exact same isotype, but it only binds to human EGFR. It does not bind to anything of the murine origin. And we use a totally murine model, the commonly used 41 murine breast cancer model. So there's nothing for EGFR to bind specifically to. So that's a perfect control to look at the API effect, look at the leaky tumor vasculature. We have labeled TRC105 with a variety of different isotopes. I'll just show you the zirconium data. Basically, it's one of the, I think we all agree that it's one of the best isotopes for labeling of antibodies, especially for clinical translation because of the good decay half-life. The labeling is quite straightforward. We just use a commercially available desferol. We didn't do any site-specific conjugation, just kind of random labeling, but we only label a few of desferol per antibody. And we make sure that it did not affect the binding affinity. After that one-step labeling, we get this zirconium label TRC105. After intravenous injection, you can see the tumor very, very well at about 1 day post-injection. The tumor is the brightest organ in the whole body. At 24 hours, you can see some signal in the blood pool in the heart. While 4 days later, you can still see the tumor, but the background has become much lower. So this is a really good contrast. So over the last several years, we have done a lot of work on TRC105. For example, we can use TRC105-based immunopad for imaging different kind of solid tumor types because they all have angiogenesis ongoing. They all express high level of CD105 during the exponential growth stage. Not only can we use this tracer for imaging tumor angiogenesis, we can also apply this in cardiovascular diseases to image angiogenesis. After disease, for example, during the remodeling process of myocardial infarction, there's high level of angiogenesis ongoing, so there's also high expression of CD105. So we can use the exact same tracer to image angiogenesis in different diseases like peripheral artery disease, myocardial infarction, abdominal aortic aneurysm. If the imaging contrast looks this good, of course, if you switch that to a therapeutic isotope, say lutetium or yttrium, that also gives you a very good therapeutic effect. We can also use imaging as a tool to monitor the treatment effect. We can give a drug to stimulate angiogenesis or use that to inhibit angiogenesis, and we can use imaging as a tool to monitor the therapeutic effect. So we've done all of this over the last several years. This is one side project that we did a few years ago. We collaborated with a group, Best Myron's group, where they want to do a stem cell tracking with reporter genes, and they chose a PET-MR reporter gene. And this, they use a divalent metal transporter as a reporter gene. They transfect that into a human neuroprogenitor stem cells. And so this divalent metal transporter can transport 2+ metals. If you use manganese, if there's high enough level, it can give you MR contrast. And we collaborated with a cyclone group of University of Wisconsin. They can make all kinds of different isotopes. One of them is manganese 52. That's also a 2+. So that you can use that for PET imaging. So that's in vitro data, in vivo data. That's in vivo MR imaging. That's a PET imaging. PET, of course, gives you a little bit lower resolution. But nonetheless, you see the data on this side when the cells were transfected with divalent metal transporter, but not on the other side. So showing that it's specific only for the cells that can transport those manganese iron. So we thought that the manganese has about a 5-day half-life. So we thought that's really a good isotope for radiolabeling of antibodies. So we tried the exact same thing using TRC105 as a targeting ligand. This is also the same same model. You can see the tumor uptake is also quite high. And it's quite heterogeneous because angiogenesis is definitely not a homogeneous process in the tumor. For PET imaging, typically what you are imaging is actually only the isotope. You may not image, if the labeling is not very stable, the isotope falls off, the data may not represent the antibody location. So we did some control experiments, and we injected free manganese chloride, so no labeling. And we want to make sure there's no signal in the tumor, and that's indeed what we found. That's suggesting that the signal here is indeed manganese linked to the TRC105 antibody. And I think that's kind of what we are expecting. But what was really unexpected was that the signal in the pancreas was quite high. So we thought that could be a really interesting, maybe we can do something about this. And in the United States, there are about 30 million people affected by diabetes, and there has been a lot of imaging agents developed for diabetes. But I think there's still room for improvement. So we thought maybe we can use this, because manganese That's a whole body biodistribution. Pancreas has the highest signal in the whole body. So this should be good for imaging diabetes. Hopefully it can be used for imaging diabetes. And the key is that for the holy grail of diabetes imaging is that you want to image the functional beta cell mass in vivo, and ideally you can quantify it. So we test this in cells. Here it shows the mechanism of manganese uptake and its correlation with the beta cell function. And this the uptake of this manganese into the beta cells is due to the voltage-dependent calcium channel, which is the VDCC. So when the beta cells are active, it takes up a lot of calcium, and if manganese is close by, it can take up manganese into the cell as well. And if we apply some stimulator that can stimulate the activity of beta cells, like the ones showing blue, it can increase uptake in the cell. If we apply some inhibitor that can inhibit the beta cell function, it can reduce uptake of manganese. So we test that in cells. Everything worked like we planned. We also tested in vivo. Here we show that the baseline, you can see the pancreas has high signal. When we apply the agent that can inhibit the beta cell function, the signal in the pancreas is much lower. When we apply another agent that can stimulate the beta cell function, the signal is much higher. So that's also that's kind of exactly like what we found in the in vitro studies. We also moved one step further into the diabetes models. This is a type 1 diabetes model that's induced by a small molecule called STZ. We can measure the blood glucose level, the BGL. After a few days, you'll see the blood glucose level is above the background. This is considered as a diabetic. So about 1 week after we did the induction, we have the diabetes model. Here it shows a baseline of the normal group, which is just like this, while this is a STZ diabetic mouse. This is a one slice, while this is a maximum intensity projection image. You can clearly see the signal in the pancreas are all gone. And we can also do staining, insulin staining to count under the microscope how many beta cells, how many islets are there. And also we can quantify it, and indeed it's much lower in the STZ group. We also did it in other diabetes models, so indicating that indeed the uptake of this manganese into the beta cells is through the beta cell function, is through the calcium channel. We also have in vitro studies to prove that. So that's manganese 52. You can it has about 6-day half-life. You can do detailed studies in animal models, but it's hard for clinical translation because you don't want that long exposure of the radiation to patients if you want to translate this. So this, luckily, our collaborator, the cyclone group led by Todd Barnhart, Jerry Nichols, and John Engel, they can make manganese 51, which has a half-life of 46 minutes. So that's really good because it's a small molecule, so the clearance is really fast. So it's 46 minutes is good enough. Here it shows a 1 hour post-injection. We can clearly see the pancreas. So the pancreas signal is as high as all the organs except the kidney, which is a clearance organ. Even in the mouse, we can clearly tell the pancreas from the kidney, and in humans, of course, there should be no problem. So currently, we're doing the paperwork for clinical studies. So hopefully, we can translate that into the patient either later this year or maybe early next year. So that's a few studies about TRC-105 imaging, which led to a kind of a side project on diabetes. We have also worked on a few other targets, for example, CD146. For this, we developed a monoclonal antibody in collaboration with a group in China against CD146. And the antibody we developed can recognize a 3D biologically active antigen rather than the typically when we develop antibody, pick a short peptide sequence, use that as antigen. The antibody usually does not work in vivo. It only recognizes the denatured protein, so it's probably good for Western, but not good for in vivo applications. And here, what we did, it's good for in vivo applications. So basically, that's roughly the procedure. The key difference is only comparing to conventional method, there are only two major differences. One is that for the antigen, we did not pick a peptide fragment. We just use the total protein. That's the extracellular domain of the whole protein. So now the antibody can recognize the whole biologically active form. The other one is that we get the activated B cells from the lymph nodes close to the site of injection rather than normally get it from the spleen. That's the two major difference. But with that, we can make it much faster and also make it low cost. In the laboratory setting, we can get gram quantities of purified monoclonal antibodies for all kinds of in vivo studies in animal models. And usually you can label it with copper 64, we can label it with zirconium 89, we can also label it fluorescent dye for image-guided surgery. And here shows the xenograft models. You can see the tumor uptake really high. When it has high CD146 expression, we can do blocking studies, while PC3 does not express high level of CD146, so the signal is quite low. That's xenograft models. This is the orthotopic brain tumor model. Here it shows the mouse, shows the signal in the mouse brain, the yellow-orange spot. This is a whole body image, not an image of just a mouse. Basically, we increase the minimum threshold, and until we cannot see anything else, and all that's left is a brain tumor. So the brain tumor uptake is the highest in this model, so it gives you really good contrast. We can also confirm the presence of the brain tumor in the mouse by MR, by H&E, and also by staining. And here it shows that CD146 expression is high in the tumor tissue, but it's very low in the normal brain tissue. We have also done a near-infrared fluorescence imaging. Basically, you can potentially use this for image-guided surgery. And here it shows the in vivo image. The signal at 48 hours is lower comparing to the absolute signal at early time points. But then if we lower, because I showed this all at the same scale, if we lower the maximum threshold, we can see the contrast is actually better comparing to early time points, because at that time, the non-specific binding has all cleared. Here it shows the ex vivo data. You can clearly see the brain tumor signal is higher than all the normal organs, including the liver. We can slice the brain in half. You can see the signal here. To confirm that indeed this antibody could potentially work for clinical studies, so we also collaborate with a group in China. We can stain the human tumor tissue that was resected from patients. And here it shows different grade of brain tumors. Generally speaking, the higher the grade, the stronger the expression. And we also, because when you're imaging brain tumor, you always worry about the blood-brain barrier penetration. In GBM, it's already broken, of course, but in the lower grade, it's not. So we test a few other solid tumor types as well, so we don't have to worry about the blood-brain barrier. So again, this is a human resected tumor tissue. In gastric cancer, ovarian cancer, liver cancer, and lung cancer, you'll see the higher grade, it has high expression, while lower grade has low expression. So this is also a broad spectrum target. While this is mostly on the tumor cells, some are also on tumor vasculature. So if we combine that with the CD105 study that I've shown you before, that one is a broad spectrum target on the vasculature, while this is a broad spectrum target on mostly on the tumor cells. So if we combine those two together, we can cover pretty broad spectrum of the tumor microenvironment. So over the last several years, we have done studies of different kind of targets, including angiogenesis, some other overexpressed targets, as well as immunotherapy-related targets like PD-1 or PD-L1. For the targeting ligand, we typically use biological molecules, all the way from peptides to intact antibodies, as well as some antibody fragments. Just to show you one example about the imaging immunotherapy. Here we use a kind of a more artificial humanized mouse models. Basically, we have the NSG mice. We implant the tumor first, and then in our UW humanized mouse core facility, we can inject the human peripheral blood lymphocytes intravenously into the mouse. So that'll give you a certain time window for the imaging, because the antibody we use against human antigen, the clinically used approved antibody. And we can use, for example, the column labeled nivolumab to image the PD-1 expressed. PD-1 on the T cells, when the T cells home to the tumor. Here shows a mouse with the human peripheral blood lymphocytes. We can see some infiltration of the T cells to the tumor, while in the NSG mice, there's no human PD-1. There's very little signal in the tumor. Of course, there's always some API effect because we use antibodies. When we do the staining, it's probably hard to see, but when we stain for CD3, which is a marker for the T cells, and for PD-1, which is the target we are imaging, they correlate quite well in the PBL mice, but not in the NSG mice, to confirm that it is indeed quite specific. We can also do on dual targeting, because cancer is such a heterogeneous disease. You can't expect to target one target and then expect to cure the cancer. In this case, we link the cetuximab Fab, which binds to the EGFR on the human cancer cells, TRC105 Fab, which binds to CD105 on the tumor vasculature. We link those two together with click chemistry. Now we have a bispecific Fab2 fragment. When we radio label that with copper 64, you can see the tumor uptake is much higher compared to all the normal tissue, all the other control groups. This is the fluorescence imaging. This is the fluorescence image-guided surgery. The tumor is quite small. It's only 1 to 2 millimeters in diameter. We can remove the tumor and then scan the mouse again to make sure there's no residual tumor tissue left in the original site. So over the last 10 years also, we have worked on a variety of different diseases. We're not really focused on cancer, although our main threat in the lab is cancer, but we did work on a variety of other diseases. For example, those are all shown here. And diabetes is the first thing that we're trying, that's the first tracer that we have developed at the University of Wisconsin that's moving to clinical studies. So that's kind of exciting times, and also it's kind of a terrifying times. So next I'll spend 10 minutes on the nanobiotechnology work, some of the work that we did recently. There are many different nanoparticles. That's a review we wrote in JNM a couple of years ago. There are many different nanoparticles that could be used for imaging therapy or theranostic applications, including organic, inorganic, and many other types. And for us, our nanoparticle-based work is mainly divided into several different kind of areas. One is tumor targeting with nanoparticles, and we chose to target the tumor vasculature because most of the nanoparticles, they either do not extravasate or they do not extravasate very well. So we chose to target tumor vasculature. So this way, when you do imaging, you don't have to worry about whether the nanoparticles can get out of the vessel or not. It works across many different nanoparticles, and we have 3 different systems for this kind of targeting. We can use RGD peptide to target integrin alpha beta 3. We can use the TRC105 to target CD105. We can also use VEGF to target VEGFR. So what we've been trying to do is that we try to maximize the EPI effect by modifying the nanoparticle surface so it has certain circulation lifetime. It also has try to reduce the positive or negative charge, so we maximize the EPI effect. And on top of that, we add the vasculature targeting that can typically enhance the tumor uptake, the absolute tumor uptake by 2 to 3-fold. In some cases, it could be even higher. So that's one slide summarizing our 10 years of work on nanoparticle-based targeting. Basically, everything we did was mostly focused on tumor vasculature targeting. The other area that we spend a lot of effort on is on new ways of labeling nanoparticles. So hopefully it can get translated into the clinic a little bit easier, because normally when you label nanoparticles, you need to label with non-lived isotopes, say copper or zirconium. So it does require some chemistry, so it's hard to make it GMP. So this is something we call intrinsically radiolabeled nanomaterials. So we pick the right nanomaterial with the right isotope. We can label it very easily. So hopefully that can have a better chance of clinical translation. There are many different ways of doing this, but we mostly focus on this something I call specific trapping. For example, I'll just show you one example. Zirconium, typically we label it with desfluor, which is based on oxygen. We have also worked a lot on mesoporous silicon nanoparticles. That's a silicon dioxide. There are a lot of oxygen in that. So we thought maybe this oxygen can be used for chelating zirconium without doing any desfluor conjugation. And indeed, it worked actually very well. And here it shows a meso channel. So the mesoporous silicon nanoparticles has a pore size of 2 to 3 nanometers. So most of the oxygen are inside the pores. And there are a lot of oxygen to coordinate with zirconium. And when zirconium goes inside the pores, it's protected from the outside environment, so it doesn't get detached easily. And we can scan the same group for mice for up to 6 half-lives. So we scan the same group for 21 days, and then we barely see any detachment of zirconium, because once zirconium detaches, it goes to the bone, and we really don't see much bone uptake. So it's very simple labeling, but it's very stable. Of course, the labeling is time-dependent, concentration-dependent, and also kind of affected by temperature. The other area we have been working on is that for nanoparticles, typically we have always used PET as one of the key imaging techniques for that because it's quantitative, it's kind of clinically translatable. So this is a review we wrote last year talking about combination of PET and nanotechnology. I think those two, when you use it together, it's really a powerful duo that can be really synergistic, not only for imaging or therapy, but also you can integrate both imaging and the therapy. I'll just show you one example that we did. This is a hollow mesoporous silicon nanoparticles. So it has a pore size of 2 to 3 nanometers, but the total size but there's a cavity in the middle, and which can serve as a reservoir for labeling those, for loading the drugs. We can link it with zirconium. In this case, we did the zirconium labeling in the first step, and then we can carry it on all the way to the end without losing any zirconium. At least we cannot measure any loss. So that's a first step with the labeling, and this is a positively charged. We can when we functionalize with amine groups, we can make copper sulfide that's coated with citrate that's negatively charged. And the copper sulfide can be used for photoacoustic imaging and photothermal therapy. We can also load it with TCPP, which is a dye that's used for photodynamic therapy. At the very end, we link the pack. We use a 10K pack to increase the biocompatibility and circulation. So in this case, we did not do any targeting. So that nanoparticle can be used for multimodality imaging and also image-guided combination therapy. So imaging, we can image with PET, which detects zirconium. We can do fluorescence, that's detecting the photodynamic dye, the TCPP. Cherenkov luminescence basically is the optical signal of the zirconium. And we can also use the Cherenkov signal to excite TCPP. That's a CRET, Cherenkov resonance energy transfer. So that can tell you whether those two are close to each other or whether they are separated apart. That can give you some information about how stable it is in vivo. For the treatment, we can use photothermal therapy, which uses copper sulfide. We can also use photodynamic therapy, which uses TCPP. And here you the TEM structures. The hollow MSN nanoparticles is about 120 nanometers in size. And each of the dot is copper sulfide. And we can use different laser for those treatment. 980 for photothermal, 660 for photodynamic. And here shows the imaging of the 4 different methods. And when we do the treatment, when we combine those two, if we just use each of the treatments, say photothermal therapy or photodynamic therapy, depending on how much energy you use, each of them can, in this case, we chose a level that can only stop the tumor growth, but it does not shrink the tumor. But when we combine those together, it can shrink the tumor very quickly. We also calculate the synergistic effect. So it's indeed 1 + 1 is greater than 2, it's not additive. It's much more than additive. Here shows the data. And all the groups, all the mice survived. Here shows the size and H&E staining. To summarize where we are and where we are going in the future, at the University of Wisconsin, one of our biggest strengths, our best collaborator is the Cyctron group. They made a lot of different isotopes, actually more than 100 over the last 4 decades. So that's our kind of a biggest gold mine that we can test a variety of different nanomaterials, different isotopes for different applications. So my PhD was in chemistry, so in our group we can do some chemistry. We can also do some biology. I have students from medical physics, so we can do some modeling data analysis for radiotherapy. We can also do some surgery to generate some disease models that's beyond xenograft models. We need people that can independently write a lot of different things, protocols, manuscripts, grants, and this and that. We can also make different kind of nanoparticles. Some we make it ourselves. We also have collaborations all over the world. They can make nanoparticles and they ship it to us. We can help them to do the imaging. We can also engineer and do targeting ligands or do nanomaterials. So what's holding all this together, you can say it's molecular imaging because it's really it takes a village to get the thing done properly. You can also say it's nanobiotechnology because that requires all of this. But in real life, I think you all agree with me that's actually funding that's keeping all this together. We're still alive for this at this point, so hopefully we'll be alive for as long as we could. So to summarize, we have developed different kind of imaging agents for imaging and therapy, theranostic applications. And I think intrinsically radiolabeled nanoparticles is really a very important method. And actually one of my postdocs, Fen Chen, is now working with Michel Bradbury. He just did this zirconium labeled CDOS. I think they're moving to clinical studies using the intrinsically radiolabeled methods. So that's really exciting. And of course, different imaging techniques, they can be synergistic and also, at least if you use them right, it should at least be complementary. So I need to thank a lot of people, our collaborators, the Cyctron group, my former supervisors, my collaborators, the funding agencies, and the companies that we collaborate with. And these are the people that did the work. I didn't do any of those work myself. My group, they have different kind of background. In the US, we talk about diversity. People in my group, they have some are biologists, some are physicists, some are chemists, some are material scientists, some are surgeons, and some have other background. They also have different ethnic background. In this photo, about 1/3 were born in the United States, 1/3 were born in China. But the other third came from different countries, for example, Cuba, India, Thailand, Brazil, and Korea. Even though they have different scientific background and different ethnic background, we work together as a group. We try to help each other because it does take a village to get things done properly. So we work together just like a family. Not only we're like a family, 2 of my graduate students, one from India, one from China, last year they just became a family. And they are, so I'll stop my talk here. I'll be happy to answer questions. Thanks.
Thank you, Weibo, for an excellent presentation. Indeed, precision
Thanks.
medicine. This presentation is open for question. Jason?
Good to see you, Jason.
Oh,
If
good
you
to see you
could
too.
use
Hey,
the
Weibo. Thank you very much.
Oh, thank you.
The first zirconium particle patient was done 4 hours ago.
Friend
So,
told me about that.
Yeah,
Yeah.
happened today. That's not my question. So the magnesium work, is it magnesium chloride that you're injecting?
Oh, that's manganese.
Manganese, sorry.
Mn. Yeah.
Jet lag, manganese. Is it magnesium chloride?
Yeah, manganese
Have you
chloride.
tried other salts at all?
Other salts like
Other salts, sulfate or anything else like that at all?
We haven't. The stuff we injected just manganese chloride. We did not. I would expect it to be the same, although we didn't try it.
No, I just know, for say, the copper studies, copper chloride and copper sulfate.
Oh, they might
actually,
behave
there might be
differently,
some
right?
difference. Have you thought about, because this is obviously calcium channel mimic.
Yeah.
So cancer, that's obviously a big issue sometimes. Have you just done magnesium chloride in a cancer model, particularly prostate?
Well, copper works reasonably well
Copper
in
does, but
prostate.
manganese, with the calcium channels and dysregulation associated
Well, at least in the
with
41 we
yeah.
tested, it didn't show any uptake in
Didn't
the
show.
tumor. We did not test across different types. It might be some cancer.
in
have.
prostate and weight. And how about in cardiac disease? Because they also get calcium channel
Absolutely,
blockers.
the hard wall has high uptake
Yeah.
for sure. Yeah, because that takes up a lot of calcium. So when we did the BioD, the hard wall does have quite high uptake.
So patients who are hypertensive who get a calcium blocker, you might be able to see if it's actually working by just giving a simple magnesium scan.
Oh, we did calcium blocking. We did the inhibitor stimulator. Those are all calcium. I mean, they are blockers of different mechanisms. Some
Yeah.
are ATP blockers, some are calcium channel blockers, some are I forgot what's the other one. But yeah, definitely, I think we are very positive it's due to the calcium channel uptake.
Terrific. Okay, thanks.
Thanks.
One last question.
Hi, Michelle, King's College London. I was just wondering about your bispecific antibodies. Do you know why you're getting higher tumour uptake? Is it a pharmacokinetic and clearance thing, or is it a sync thing, or is it a divalent binding?
I mean, it's
Side
definitely
effect?
a mixture of different things. One is that it's when you have the Fab2, it's bigger than Fab, so that can have a
Yeah.
longer circulation lifetime. So that's part of it.
Yeah.
It could be binding 2 things at the same time because we have 8 ethylene glycol chains
Yes.
units, so it's possible. But maybe most likely it's kind of a local high concentration effect. So one, because the binding is dynamic, if one falls off, the other one can kick in right away, rather than losing it. So I think it's a mix of all of this.
How does it compare to, say, your same, I guess,
Fab2?
dimer? Yeah, your Fab2 fragments with a homo sort of Fab.
In this particular
Combination.
study, we just compare with Fab, but in a subsequent study, we compare with the Fab2. Actually, those are higher than both Fab2s. Although the exact mechanism I still don't know because we don't have the
Yeah.
expertise to tease that out exactly what happened.
Okay. Thank you very much.
Thanks.
Great. Thank you, Weibo, for an
Thank you.
excellent presentation.